Welcome, everyone, to today's session of our Now Program educational webinars. Today's topic will be oncolytic viruses for brain tumors. This program is hosted by the Blum Resource Center. Over at the Blum, we are grateful to the Now team for this continued partnership. The recording of this session will be posted on our Blum YouTube playlist in our Blum Digital Resource Center. For those of you watching live, if you have any questions related to today's program, please feel free to utilize the chat and Q&A feature throughout the session. At the end of the session, your questions in the chat will be used to moderate a Q&A. If you have any other questions or would like more information about resources and programs offered through the Blum, please visit our website at www.dana-farber.org/resource center or email us at blum_center@dfci.harvard.edu. Thank you all for joining us. I will now pass the workshop over to Dr. Gonzalez Castro to introduce our panelist. Thank you.
Thank you so much, Sierra, and thank you to Rachel Fox, our administrative director, for all the work behind the scenes to make the webinar happen. Happy New Year, everyone, and good afternoon to all those on the East Coast. Welcome to another edition of our Neuro-Oncology Wellness Education webinars. As you know, the Neuro-Oncology Wellness Program is an initiative from the Center for Neuro-Oncology at the Dana-Farber Cancer Institute and Brigham and Women's Hospital, with a mission to educate patients and their caregivers about brain tumors, as well as provide resources to promote overall health and well-being. My name is Nick Gonzalez Castro, and I'm the director of the Neuro-Oncology Wellness Program. And today I'm joined by Dr. Nino Kioka for a discussion on targeted therapy and oncolytic viral therapy for gliomas. Dr. Kioka is the Harvard Cushing Professor of Neurosurgery at Harvard Medical School, as well as the Chair of Neurosurgery at Mass General Brigham, and the Executive Director of the Center for Tumors of the Nervous System at Mass General Brigham, where he also serves as co-chair of the Executive Committee of the Cancer Institute. In addition, Dr. Kioka is a world-renowned authority on oncolytic viral therapy for glioblastoma and other aggressive gliomas, leading a cutting-edge lab at the Brigham and Women's Hospital, and he has authored over 450 peer-reviewed publications, and he's also the recipient of numerous awards. He has served as president of the Society for Neuro-Oncology, the American Academy of Neurological Surgery, and the Society for Neurological Surgeons. More importantly, he's a dear colleague, and he has been caring for brain tumor patients for over 30 years. And his career has been devoted to finding more effective treatments for patients with aggressive brain tumors such as glioblastoma. So join me in welcoming Dr. Kioka. Thank you so much for joining us today.
Thank you. Thank you, Nick.
Before we start, I want to remind the live audience that we want you to be part of this conversation by submitting your questions via the Q&A feature at the bottom of the Zoom screen. And we will select a number of questions for our guest, Dr. Kioka, to answer in the second half of the program. The program will also be recorded and will be available at our website now.dana-farber.org and also on the Dana-Farber YouTube channel. Our discussion today will be centered on the use of viruses to treat tumors. So let's just start at the very beginning and help orient our viewers today. What is a virus, Dr. Kioka?
Yeah, we think of viruses. Well, first of all, thank you. And it's great to be here. So we think of viruses as pathogens, and clearly with the recent pandemic with coronavirus, we know that viruses can cause disease and illnesses. Viruses like Ebola virus and HIV, for example, have been well-known scourges to humankind over the last few decades. And so we think of viruses as pathogens. But viruses really are very efficient structures that can deliver genes and nucleic acids into mammalian cells. And as they do that, they can replicate their genetic material and create progeny. And this is the pathogenic mechanism. Now, we and others have wanted to change the ability of viruses to do this so that instead of delivering just their own genetic material, they can deliver additional genetic material to either make the cell, particularly like a cancer cell, into something that dies or is or is killed, or something that maybe promotes immunotherapy and immunologic responses.
Wonderful. Thank you for that very detailed and informative answer. You mentioned a number of different viruses, Ebola, HIV. To be more specific, what viruses have been used or are being used to treat brain tumors?
Yeah, there actually a variety. The most common one has been a virus based on herpes simplex virus type 1, which causes the common uh, uh, labial oral mucosal lesions. Um, and it's present in about 2/3 of the human population. But other viruses have been modified to also kill tumor cells, like brain tumor cells, such as adenoviruses that cause the common cold. Poliovirus, they used to cause paralysis, measles as well, retroviruses. So there's a lot of different viruses that one can modify in the laboratory to make them safe for normal cells, but deadly for cancer cells.
I see, and I want to expand on that because, as you noted, HSV can cause oral lesions as well as brain infection, and then poliovirus can cause paralysis. So, if these viruses have a normal tendency to attack nervous system structures, how is it that we can then harness them to target tumor cells and not really cause disease?
Yeah, no, so there's, with the wonders of genomics and molecular engineering and technical genetic engineering, we can now modify these viruses so that they can be selective for tumor cells and only target tumor cells either in the process of entering the cell to specific receptors. So you can modify the virus so it'll only enter tumor cells because those tumor cells will have the loading dock to allow the virus in while normal cells do not. Or once that virus is in the cell, the virus can survive and make copies of itself only in the tumor cell, but not in the normal cell. So those have been the primary mechanism which we've been able to make these viruses selective for tumors versus, let's say, brain cells.
I see. So these are viruses that share most of the features of the normal virus that can affect and cause disease, but they have been specifically modified not to do so, and perhaps further modified to attack cancer. Earlier, you mentioned poliovirus, and a few years ago, there was a lot of enthusiasm on the basis of tumor responses seen in patients treated with a modified poliovirus. And there have been a couple of studies on this. Can you discuss, what is the state of poliovirus for the treatment of brain tumors?
Yeah, so poliovirus is very interesting because they modified the virus that will only attack tumor cells and not normal cells. So that's the way they modified that. And there was some very promising clinical trial data in glioblastoma patients where they treated over 60 glioblastoma patients and showed a number of patients that had some very positive responses. They've gone into a phase 2 trial, which enrolled more patients. That data has not been published yet, so I don't know what those results were, but at least the phase 1 results seem to be very encouraging. But like most of these things, we need to get further validation to know if this is going to be a drug that's going to be approved for regular use for all patients with brain tumors.
Thank you. Thank you for providing an update on that. Your laboratory has developed a different approach to treat brain tumors using the herpes simplex virus 1 or HSV1. Can you tell us more about the rationale for using HSV1?
Yeah, so we've been very interested in HSV1 because it likes to infect cells in the nervous system. So since glioblastoma arises in the nervous system, we thought that this would be a good virus to work on. It also has the ability to easily modify a lot of its genetic elements. It is what's called a DNA virus, which allows us to use just regular molecular biology techniques to modify it. And there have been several herpes simplex viruses that have been in clinical trials for glioblastoma. Each one is a little bit different. It's like using different drugs. And so the one we've worked in as certain parameters that allows it to selectively, not just in fact, but once it's in the cell, selectively grow in the tumor cell. But if it infects a normal cell, it will not grow. And this particular herpes virus not only does that, but also sets up a very profound immune response in the patients. And we think that is a major mechanism that allows the host, the patient, to reject the tumor as it's trying to reject the virus as well.
Very interesting. Let's get a little more into the specifics of how this therapy is implemented. How is this HSV-1 virus delivered to the tumor, to target the tumor?
Yeah, so at this point, the way we're delivering this is just with a needle inside the tumor. So the patient does require a small incision through the brain. It's actually not through the brain, through the skull. And then we place a very small, tiny needle into the tumor and then inject the virus slowly inside the tumor. It's almost like getting an intravenous injection inside your tumor. We use some sophisticated tracking devices to make sure the needle is really in the tumor as we do this. And so it's called an intratumoral administration.
Very interesting. Very interesting. And do you inject one area, the largest area that you notice, or you try to go for different sites? Sometimes we see in our patients that the tumors are somewhat more diffuse.
Yeah. So actually what we've been doing is we've been doing testing in multiple ways. One has been to just inject one area, the area that looks most malignant. Another way is to inject multiple areas. In fact, we've injected up to 5 areas in the tumor. We're now testing injecting at multiple time points, up to 6 time points over 4 months. Because as these tumors evolve, we wanted to hit a new area of tumor evolution or tumor formation with the virus. And so we're testing all those administration modalities to see which one is most effective.
How soon are repeated tumor injections given to patients?
Yeah, so in the current trial, we're giving an injection 15 days after the first injection, 30 days after the first injection, and every month, up to 4 months. So it's that for a total of 6 injections.
I see. And then these patients are monitored in the hospital, or are they soon discharged?
Yeah, so
home?
we're trying to do this as an outpatient procedure as possible. The patients they're asleep, but they're not under general anesthesia. I explain it's like getting a biopsy of a breast biopsy or colonoscopy, for example, where they give the anesthesiologist gives you drug to make you doze off while we do this, but you're not under anesthetic. We're trying to get patients out either the next morning. I've had some patients actually go home the same day. It's an outpatient procedure. And we're using very small incisions sometimes because all of these patients are at the time of recurrence. In other words, the first tumor was treated, but then it came back. So that's when we're treating patients. We're using the same small holes in the skull that were placed in the first operation to inject the virus. So there isn't a lot of surgical maneuvering that we have to do to get to the tumor.
I see. And these patients are then subsequently imaged for response. How soon are responses seen, and what are things that could be found in MRIs shortly after treatment?
Yeah, so the one problem we found is that the MRI scan really measures tumors, but it uses parameter surrogates or other modalities to say this is a tumor, but they're not actually tumor. It's just the fact we're measuring blood vessels and the permeability of blood vessels in tumor and say that is tumor. So it works. MRI scan really work really well for drugs. chemotherapies. The problem with this therapy is that when we inject the virus, that also changes the permeability of blood vessels. And so sometimes it looks almost like the tumor is growing, even if we inject the virus. But then when we go back, what we see is not that the tumor is growing, it's just that the tumor cells are dying and they're recruiting a lot of immune cells into the tumor. They're fighting off the tumor. And that's giving this paradoxical increase in what we call tumor, but it's not really tumor.
I see.
That
I
makes
see.
sense. Yeah.
So is that cell death and associated inflammation,
Yes.
does it often lead to symptoms in patients?
Sometimes, especially if it's very close to areas of the brain that are involved in motion, vision, or sensation, or speech. And so, we've had to sometimes uh, play around with steroid medication to reduce that. The problem with steroids is it can also reduce the immune effects that we're trying to provoke. We've worked on using some low level of what's called bevacizumab, which is a drug that also reduces swelling, to try to improve that. And the nice thing about bevacizumab is that it does not change the immune system or the immune response to the tumor.
Yeah. Yeah, it's a difficult problem. We definitely want to benefit from that inflammation and the
Yeah.
recruitment of immune cells to further help clear tumor, while at the same time managing symptoms. Related to this, last year you published the results of a phase 1 study using this virus. And the study showed that the treatment was well tolerated and you saw an encouraging number of responses. Um, the can you summarize the study? And also, I think there was something very interesting in relationship to prior exposure to HSV1. You mentioned that a high percentage of the population are seropositive. So just take a few minutes to walk us through that.
Yeah, so this is a study, where we first treated patients that had a recurrence of this tumor. A recurrence, there's not really a very good treatment. So this is why they were eligible for this clinical trial. And we injected the tumor with this particular oncolytic herpes virus. And what we saw was the following. Uh, uh, in several patients, we saw a lot of inflammation, actually the majority of patients. When we looked at the tumor, the inflammation was due a lot to the tumor-fighting immune cells, which are called T cells.
Mm.
We saw a profound infiltration of these tumor-fighting immune cells coming into the tumor. And we could actually correlate that the amount or the changes, the more T cells came into the tumor, the better the patient survived, the more the patient survived. And this is particularly true in the patients that had been previously exposed to herpes simplex. So patients that had pre-existent or previous exposure to herpes seem to be the ones that have the most T cells coming in. And that way you think of this is almost like when you get a vaccine, the first dose of a vaccine does not work well. And that would have been your first exposure ever to herpes because you caught it, out in the general population. With a second or third exposure to the vaccine, that's when your immune system really gets revved up. And so with this injection, we were giving people a second dose of this vaccine. So we could correlate the more T cells coming in there, the better the response. T cells are interesting because each one of them is a separate signature. It's like they have a separate zip code. And the zip code of the T cell tells you a little bit what it does. And so we could actually correlate some of the zip codes of these T cells with the responses that we saw. And now what we're trying to do is with these multiple injection trials is to really get a timeline of this process. This was just looking at one time point, the one I told you about. But with this multiple injection trial, now we can look at the timeline of these T cells coming in there after injection of the virus and study how these T cells start destroying the tumor or not destroying the tumor. If they're not destroying the tumor, we'll figure out why that is so.
Very, very interesting. We have discussed in previous webinars on the topic of immunotherapy, the use of drugs called immune checkpoint inhibitors to try to further activate T cells to clear tumor cells. Based on the results of your study so far, do you think this is a strategy that could be pursued of combining immune checkpoint inhibitors with the oncolytic virus?
Most definitely. And I think that is something we want to do. We have not been able to do it yet. In a few patients where, that lived a little bit longer, and then we wanted to add something else, we added standard of care immune checkpoint inhibitors, and it worked fairly well. But we've even done that two things at the same time together. We've also wanted to do the virus together with peptide vaccines. I think this is going to work the best in combination with something else.
Interesting, very interesting. And just to clarify for the audience, peptide vaccines are vaccines that are designed to target a specific molecule or protein that is present in tumor cells. So to generate a population of these T cells, and I think, as Dr. Kyoka alludes, it makes a lot of sense given the fact that the virus helps activate the immune system to combine it with the virus. Is the study still ongoing? What are the next steps?
It is. So again, we are at this, and we have done the first 6 patients where we're doing these multiple injections, and we're accruing for the next cohort of patients where we're again trying to figure out if you do multiple injections, just like when you get a vaccine every year, you have to get your flu vaccine to rev up your immune system. So we're doing the same thing with this to see whether it seems to be fairly well tolerated so far, and we're trying to finish up that part of the trial. I think the next step after that is going to be up. We'll have to figure out, we'll have to decide together with regulatory agencies.
Excellent. Is your laboratory trying to modify the HSV-1 virus further or work on other viruses for clinical trials?
I think one of the exciting things is we talked about combinations. So you can combine the virus with a systemic therapy with an immune checkpoint inhibitor, but you can also engineer these viruses so that they could express in by themselves the immune checkpoint inhibitor or some other cytokine or gene, so that they're not just killing the tumor cell, but also delivering molecules in the tumor that can make the tumor even more responsive to immunotherapy. And that is what we're doing in the laboratory now.
Wonderful, wonderful. Do you have other studies that you're planning to do then with some of these viruses?
Yes, we're going through the process now of making the virus clinical grade and applying to the FDA so that we could do a first-in-human study with these other viruses. It's a relatively long process. The regulatory process can be lengthy, appropriately so, because the FDA wants to make sure that there's safety in these viruses before they allow us to put them in humans.
Absolutely. Yes, yes, yes. Thank you for providing a wonderful overview of oncolytic viral therapy and the work of your lab. Let's take now a few minutes to answer questions from the audience. And we have a couple now in the Q&A section. And again, I encourage any viewers to write their questions there. So the first question is, are all the clinical trials in GBM, I guess oncolytic virus trials in GBM, or are there any available for other brain cancers? Particularly, this viewer is interested in high-grade glioma that is not GBM.
Yeah, so actually the trial I told you about was in high-grade glioma. So we had mostly GBM, but there were also some IDH mutant grade 3, grade 4 astrocytomas, as well as some grade 3 oligos, although it was a very small percentage of those patients. And we examined them separately from the GBM group. A lot of the, but now this multiple injection trial I was telling you about is just for GBM because we wanted to keep this a fairly pure patient population. For some of the other oncolytic viruses like polio, they also were treating this heterogeneous population, not just GBM, but also high-grade gliomas, for example. So it does work for, well, at least the trials are designed for all high-grade gliomas.
Wonderful. The second question is a related question, and perhaps you answered it already, but is what work has been done or planned to be done with oligodendroglioma? And as you mentioned, this is a type of IDH mutant glioma, and it can be high grade 2, so grade 3. So I guess those patients would be eligible for the studies that you're planning.
They are not this particular one where we're doing multiple injections, but yes, they were eligible for the other trial because then we took all of the recurrent aggregate gliomas, including the grade 3 anaplastic oligos. Not the grade 2 oligos are not are more benign, but just the grade 3s.
Wonderful. Uh, I don't see any other questions at this time, but I would
There
like
was a
to,
question I just saw about
let
whether
me see.
this is available outside of a clinical trial. Is that available? It is not. The only oncolytic virus right now, based on herpes, that's FDA approved is one called Imlygic, and it's for melanoma, not for glioblastoma. It's a separate, it's still herpes simplex virus type 1, but it is engineered separately differently and does things a little bit differently.
Thank you for noting that and answer that question from the viewer. Yes, it's important to highlight that all these therapies are still experimental, and although sometimes we use drugs that are approved for other cancers in brain tumors, off-label use is called. It is not possible to do this with this very specific form of therapy where the viruses are engineered to treat specific tumors. I wanted to ask you if you could just leave our viewers with any final comments or thoughts. You know, as I mentioned at the beginning, you've been caring for glioblastoma and other glioma patients for over 30 years, and you have watched the field evolve. So if you have any parting thoughts that you would like to share with us, we would love to hear about them.
Yeah, I think you can be very negative about this type of cancer just based on our inability to get a lot of FDA approval for drugs. I think but I think the positive thing is that there's been a lot of resources, NIH money, foundation money, industry money there to help us understand this cancer better. We're getting a lot of good information about this cancer, how it evolves. And I really feel positive that we will have some answers, even therapeutically for patients soon. Soon, it may not be months, but maybe in a few years. I do think that there's a breakthrough there that may happen. We've had one for IDH mutant gliomas recently. So I'm just
Yes.
saying that there will be one for IDH wild type gliomas as well, GBMs.
Yeah, that's our hope, and all of us in different ways are working to make this happen. But you know, we're all encouraged by the very nice progress, and I particularly have been encouraged by the very nice progress in the oncolytic viral therapy area. I just checking to see if there are any other questions. I will remind all viewers that if you want to run through all these questions again and get the information shared by Dr. Kioka again, you can access this through our website or through the Dana-Farber Blum Center YouTube channel. And we will post a survey again at the end of the program to gauge how you benefited from this webinar. And I want you to join us again next month for a discussion on the management of brain tumor symptoms with Dr. Callum Lee and Dr. John Rhee from the Center for Neuro-Oncology and the Center for Supportive Oncology. And this will be on February 21st at 2 p.m. And we'll be sending additional details shortly. But thank you so much again, Dr. Kioka, for taking the time to share your knowledge and answer live questions from our viewers. And thank you all for joining today as well.
Thank you.
Thank you both so much, Dr. Kioka and Dr. Gonzalez Castro, for that great presentation. We appreciate your partnership. As I mentioned earlier, it was recorded, and I will make sure to share the link with both the Now team where you can get it from, or it'll be posted on our Blum YouTube playlist online in a couple weeks. So thank you for that. And I will post that poll now. Thank you.
Thank
Thank
you.
you, Sierra. And please, the attendees, take a minute or so to answer the poll. This will help us for future programs.
Thank you. Goodbye.